Okay, so we have talked about how we know DNA is our hereditary material, and we have talked about DNA replication. It was a little bit complicated, these different enzymes we use, and the lagging strand and the leading strand, and the directions things are going. So I'm going to review this one more time before we go on and talk about how DNA is going to package up in your cells, and also how DNA can fix any mutations if any problems happen with this. So let's watch this little clip first.
Here you can see the simultaneous formation of the leading and lagging strands. A DNA polymerase assembles a continuous leading strand, while primase, other DNA polymerases, and ligase all work together to make the lagging strand. The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication.
Okay, so let's go through these enzymes again. Helicase is going to unwind those two strands that are together. It's going to separate them. Single strand binding proteins are going to bind onto those single strands and kind of hold it open until you start having the other enzymes coming. Topoisomerase is going to stop it from over twisting or getting tangled, so it keeps things kind of straight. It can break and swivel and rejoin DNA so that you have good DNA molecules that aren't breaking through the whole process accidentally. Then when you've got it open, primase comes in, knocks off these single-stranded binding proteins, and puts on a primer. That primer is made of RNA, and it only moves in the 5' to 3' direction. So the enzyme or the DNA strand it's working on is going 3' to 5'. What it's building is 5' to 3'. Then when you have a primer, DNA polymerase 3 attaches to that primer and starts creating DNA. It adds DNA still in that one direction, 5' to 3'. And it does that by base pairing the different bases that are on the old strand to know what base to put on the new strand. On the leading strand, it's just going to go as one long piece. On the lagging strand, though, it's moving opposite from the direction that that replication fork is open, so you're going to end up with little pieces. DNA polymerase 1 is going to come in, take out those RNA primers, and put in DNA. Because we don't want RNA mixing with our DNA. It's going to take out the RNA and put in DNA. Then on the lagging strand, all of those little Okazaki fragments are going to be joined together with DNA ligase. Okay, so all of these enzymes are working together kind of as a big clump of proteins, a big complex. So the proteins that participate in DNA replication can form this large complex called the DNA replication machine. And often, instead of it moving down DNA, they're going to stay still and the DNA is going to move through it. So that video we just watched showed how DNA can move through those complexes. So a lot of studies we've had lately show how the DNA polymerase can kind of reel in DNA, so it just kind of pulls it through it and kicks out this newly made DNA. So it's pulling the DNA through it and kicking out this newly made DNA. Although this is still pretty decently new science, we don't exactly know how it is actually, what the exact mechanism is. Okay, so then what if you have problems? DNA polymerase is able to proofread. It is going to proofread and say, oh no, there's a mistake here, and fix anything. One type of repair is mismatch repair. So when you have the wrong base put in it, mismatch repair will take out the wrong base and put in the right base. Besides just DNA polymerase accidentally putting in the wrong base and then being fixed by mismatch repair, DNA can also be damaged. So if you are exposed to chemicals or physical things that are harmful, so cigarette smoke, x-rays, sunlight, these can all damage DNA. DNA can also just spontaneously cause problems. So here's one example. You have two DNA, on DNA you have two bases next to each other that instead of base pairing with the other strand, they're pairing with each other. And when it's time to copy DNA, that would be copied incorrectly. It wouldn't be read correctly. And you'd end up with mutated DNA. Our cells tend to protect against DNA mutations. So something called a nuclease is going to cut out this broken part. So nuclei telling you it's nucleotides, and ACE telling you it's breaking it out. So that nuclease is going to break out this part of DNA that includes this, this dimer, and then DNA polymerase can come back in and put in the right thing, and then DNA ligase seals it all up. This is called nucleotide excision repair. So nucleotide, we're dealing with nucleotides, excision, which means cutting out, and repair. So a nucleotide excision repair, that nuclease cut out the damaged DNA, and it was replaced by DNA polymerase, sealed up with DNA ligase. So now you have a good, properly functioning strand of DNA, so that DNA can be copied correctly and not have any mutations in the daughter cells. So the error rate after proofreading and repair is low, but it is not zero. If there are changes, it could become a permanent change in the DNA sequence and would be able to be passed down to the next generation. Those changes are called mutations, and mutations will then, you'll have a child with a mutation that has a slightly different protein, may have a new function, may have lost a function, may have a slightly altered function. That is a source of variation that allows natural selection to choose the mutations that are most selective. And ultimately, if you get enough mutations, you can end up with a new species. One important thing when we're replicating DNA, when it gets to the end, you have this primer at the end, and then the primer gets taken off. Then what? DNA can't just keep going at the end of something. So the usual replication machinery can't complete those 5' ends. So, you know, you have a piece going this way. If you take off that primer, there is no primer for it to attach to and complete this piece of DNA. So this piece of DNA can't be copied. That means that every round of DNA replication is going to shorten your ends. You'll get shorter and shorter ends, and you'll have uneven ends because you have this kind of, this strand is there complete, and on the other, you know, on the leading strand, it's going to be finished off, but on this lagging strand, there's no way to finish that. This is only a problem in eukaryotes, not a problem in prokaryotes, because prokaryotes have circular DNA, so there isn't an end. It just goes around the circle until it meets up where it began. In eukaryotes, the end of our DNA is called telomeres, and those telomeres have specific nucleotide sequences to them. And by adding those telomeres at the end of it, it doesn't stop this process from shortening them, but it adds this extra DNA at the end of it. So when DNA starts, the ends start getting shorter and shorter, it's those telomeres that are being worn away. So your actual functional genes that you need to keep aren't getting worn out. The telomeres are getting shortened. It has been proposed that shortening of telomeres is connected to aging. So as someone gets older and older, their cells have gone through more and more DNA replication. And so those telomeres have shortened through every generation. Especially if that's a cell that's replicating very quickly, you can end up with some of those effects of aging. So here, these are our chromosomes, and this stain just stains the telomeres, and you can see these ends are lit up. That's where their telomeres are. Okay. So if in our germ cells, if I passed on shorter DNA strands to my children, they passed on shorter DNA strands to their children, they passed on shorter DNA to their children, eventually the whole chromosome is just going to wear out, right? Eventually our children are, after so many generations, that final generation, just their essential genes would be gone because those ends would have shortened too much. So even though your telomeres are shortening every time that we are replicating, that our cells are replicating, we have this enzyme called telomerase. And telomerase lengthens out those telomeres in your germ cells. So your sperm cells and your egg cells have nice long telomeres again, so that as that child develops, those telomeres start out long and can shorten. And then their egg cells and their sperm cells will lengthen out their telomeres. So every generation of people, we're not shortening our cells because telomerase is lengthening those telomeres. So for as long as I'm alive in myself, my telomeres are shortening. But in my sperm cells and my egg cells, those telomeres get lengthened again, so that my children end up with the full length into them. So that's very important. Telomerase is very important at keeping our species alive, keeping the DNA able to be passed down with integrity to the next generation. Okay, one thing that is important about telomeres is that if a cell is replicating really fast, those telomeres get worn down, and then those other genes start to be affected. So one type of cells that is replicating excessively are those cancerous tumors, right? So as those cancerous tumors keep replicating, that process of shortening your telomeres eventually are going to limit how much those cells can replicate. So it is kind of a self-protective mechanism to protect against cancerous cells. Some cancer cells are able to avoid that. They activate telomerase so that they keep those telomeres long and are able to keep replicating. So if a cancer cell is able to activate telomerase, that cancer cell is going to be able to persist longer and cause more of a problem. It won't eventually just wear itself out. Okay, so a bacterial chromosome, let's talk about bacteria specifically. They are double-stranded, circular DNA molecules, and they have a little bit of protein on them. In eukaryotes, our chromosomes are linear, they're long strands, and they're associated with a lot of proteins. In a bacteria, the DNA is supercoiled, so it's coiled up a lot. It only has that little bit of protein, so it's kind of coiling around itself, and forms the nucleoid. The nucleoid is that DNA in a bacteria, the main chromosome in a DNA, outside of a nucleus. In a eukaryotic cell, DNA is going to be carefully packed around proteins, and that protein and DNA complex is called chromatin. And they're going to go through several layers to actually be able to fold up correctly so that DNA is able to sit in your cells how it's supposed to. So chromosomes are going to go through this elaborate system, and it's multi-level. There's several steps we're going to go through. The first is that these proteins called histones. Histones are going to combine together to make these little packages that DNA can wind around. So here's your DNA, and here's this little complex of histones, and your DNA just winds around it. So you're going to form what is called a nucleosome. That is DNA wrapped around your histones, and that is 10 nanometers in diameter. I'm not going to ask you size, but it's just gradually going from this 2 nanometer DNA to now you're wrapped up to a 10 nanometer diameter. And then these nucleosomes can wrap around each other to make a 30 nanometer fiber. And then these are going to form these little loops, loop, 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 loop around it. That gets you to a 300 nanometer fiber. And then these are going to condense into the chromosome. And that final chromosome, that chromatid is 700 nanometers for one, or 1400 for their whole replicated. So it wraps around these, then these package up, then these loop, and then it condenses into the final chromosome. Okay, unfolded chromatin is going to look like beads on a string. So each nucleosome is DNA wrapped around, here is DNA wrapped around your histone. And so they have 4 basic histone types, and each one is going to be there in two pieces, so 2 of each type. So you can see it's 1, 2, 3, 4, 5, 6, 7, 8. You have those 8 little histones. So those 8 core histones. And the intermini, or the tails of the histones, are going to protrude from the nucleosome, so it just kind of sticks out. And the nucleosomes and histone tails are involved in regulation of gene expression. So if that gene is wrapped up tight, it is not going to be able to be expressed. You can't copy it to make any sort of RNA or proteins. So the way that nucleosomes are wrapping up is going to be determining if that DNA is accessible to be used by the cell to make proteins, or if that DNA is not accessible for the cell to make proteins. So this is an important way that, your eye cells are not turning on stomach acid genes, and your stomach cells are not turning on eyelash genes. Each different cell type is, although it has all the DNA, it is going to be able to control what DNA gets expressed. And one of those ways we control it is through this DNA packaging. Okay, let's watch this clip.
Several levels of DNA packing make up the compact chromosome seen in a metaphase cell. Even when a cell is not dividing, some of the DNA is folded tightly. The degree of DNA packing makes certain genes available or unavailable for transcription. Chromosomes consist of loops of supercoils, which are made up of tight helical fibers. These fibers are composed of nucleosomes, which consist of DNA wrapped around protein molecules. In the fiber, the DNA is unavailable for transcription. Each nucleosome is a cluster of proteins called histones. Nucleosomes change shape and position to allow RNA polymerases to move along the DNA and read the genes. Chemical modification of DNA and histones may affect chromatin structure and access to specific genes.
Okay. Chromatin will undergo changes in the way it's packed during the cell life cycle. So in interphase, the DNA tends to be organized into the 10 nanometer fiber or into a 30 nanometer fiber through folding and looping. And those interphase chromosomes are going to be kept in specific regions of the nucleus so that the fibers don't get entangled with each other. So it's during interphase when that cell is copying its DNA isn't staying tightly wound up. It's going to be relaxed into those 10 nanometer fibers and 30 nanometer fibers. And because all of that DNA is relaxed, you need to be able to mark it to different places. So here you can see, like, this is where you've colored each of your different chromosomes, different colors, and you can actually see where the different chromosomes are attaching to different parts within the nucleus so that they're not mixing up and tangling up with the other chromosomes. Most of your chromatin is going to be loosely packed in the nucleus during interphase, and then it's going to condense prior to mitosis. So this, let's go back. This way that we see chromosomes is only how we are going to see it during mitosis. DNA doesn't condense like that unless it is going through mitosis. And there's two different ways that DNA can be wrapped around these histones. If it is loosely wrapped, it's called euchromatin, and if it's more tightly packed, it's called heterochromatin. Heterochromatin is going to be a little more densely packed, which makes it more difficult for it to express genes. So when your cell is trying to undergo the normal things it's doing, its DNA that needs to be expressed is going to be as euchromatin. We'll talk about in chapter 17 how that DNA actually gets expressed, but it needs to be kind of less tightly packed. So this more loosely stuff is what's going to be the genes that will be able to be accessible to be read. And this more tightly packed heterochromatin, by packing it into heterochromatin, we make it more easy for the cells to be organized and it's more easy for the DNA to be organized and for those chromosomes to be separated without having tangles and twists. But it's less likely that this DNA can be read and make proteins and allow your cell to perform different functions. Histones can go through modifications that can cause the DNA to condense, and this can also have effects on gene expression. So here, if we're putting an acetyl group on your histone, that DNA is going to be accessible. But when we take it off, you can methylate your DNA. You can methylate your histones, and that will cause your DNA to be wound up more tightly. Okay, so let's go through this last summary. So DNA exists as a backbone of sugar and phosphate, and you have 4 different bases, T, A, C, and G, that are going to bind to each other with hydrogen bonds. Thymine and adenine always pair together, cytosine and guanine always pair together. So we always see equal numbers of thymine as adenine, they're always the same, and levels of cytosine and guanine are always the same. That's Stargaff's rules. Then how DNA is replicating. You have helicase opening up these 2 strands of DNA. So you have 2 different strands going 2 different ways. This is your origin of replication. Single-stranded binding proteins are going to bind to the parental DNA to keep it from zipping back together. RNA primase will put on an RNA primer on it. From there, DNA polymerase 3 can start synthesizing DNA. DNA polymerase 1 is going to take out the RNA primers and replace it with DNA. And then these strands, these short little pieces on the lagging strand, are called Okazaki fragments, and they get joined together by DNA ligase, so you have one solid piece of DNA. On the leading strand, after you put down that RNA primer, DNA polymerase 3 just goes through and makes DNA in one big piece. Okay, so that was a lot of information in that chapter. I love the stories we talk about, and how DNA is replicated is really important. So those are things that you need to be comfortable telling me, those enzymes, telling me those people and their stories. Tell me about how DNA protects itself, how DNA packages itself. you know, what form of packing you see during mitosis, what form of DNA packing you see while the cell is just existing and doing its job. These are all things you should be comfortable with. And if you have any questions, please let me know. I'm happy to help you understand things. And one more chapter, and we're done for this semester. See you later.